And our first speaker would be Warren Porter, a professor of zoology and environmental toxicology at the University of Wisconsin in Manhattan. Dr. Porter's research has shown that combinations of commonly used agricultural chemicals in concentrations that mirror levels found in groundwater can significantly influence immune and endocrine systems, as well as neurological health in animals. His recent research links pesticide exposure in utero to impaired learning, changes in brain function, and altered thyroid levels. His lab has also shown lawn chemical mixtures at low levels increase abortion rates in lab animals. Dr. Warren is a Beyond Pesticides board member, and I should add that when I first read his article, it really opened my eyes to the fact that low levels of these pesticides in combination can have a profound effect. And pesticides, has been said before, have not been tested in combination before. So this was really groundbreaking work and very important. So Dr. Porter, take it away.
Thank you, Jerry. When I first showed this to my Zoology 101 class the first time, I said, "What do you see in this picture?" And somebody in the back of the room hollered out, "Raging hormones." And unfortunately, in some cases, as we're going to see, that may be true for this little couple here where she's offering him some Halloween candy. I could subtitle this talk, instead of Lawn Pesticides, a DNA code TTCCT, which stands for the tyranny of corporate chemical trespass, a la Sandra last night. I want to explain several things in this very brief talk. One is why are environmental chemicals, pesticides especially, able to get into our bodies so easily? What I have represented right here is a cross-section through a leaf. And in order to get a pesticide into a plant to kill it, if you have a herbicide, for example, you want to get through the waxy surface, and so you add non-ionic solvents, sometimes referred to very loosely as organic soaps, but it has nothing to do with organic the way we think about it. The other way to get in is through the breathing pores of plants, through the stomates. And the lining of each one of those is a small hemispherical bulb of water, which has a lot of surface tension. But if you add surfactants, you weaken that surface tension, and so you get rapid penetration through the respiratory surfaces. This is how plants work, and unfortunately that's also how we work, because our skin is a waxy surface, and our lungs have these same kinds of surfaces here, and so the same kind of solvents and surfactants that are so well functional in terms of getting pesticides into plants promote their entry into our bodies as rapidly as they get into plants. And then once inside the body, they are bypass the natural defenses of the body. Well, now we got it in the body, now what happens? I want to talk a little bit about pesticide structure and its function. When you design a pesticide, this one here is a ring-shaped structure with a nitrogen, carbon, nitrogen, carbon, nitrogen, carbon. And what that structure does is basically, because it has very little electrostatic charge, it makes it fat-soluble or lipid-soluble. And then what's done is you add electrostatic charges, like a nitrogen and these two hydrogens, to give it some electrostatic charge. That makes it partly water-soluble. So you have a molecule that dissolves both in fats or lipids as well as in water. And why do you want to do that? Because every cell has a phospholipid cell membrane around it. And fat dissolves in fat, and so this part of the molecule allows you uniform entry into the cells of the body, any cell of the body, because every cell of the body has the same coupling. And now, once you get inside the body, that electrostatic charge is really crucial, because as anybody who's taken physics knows, opposite charges attract, and so a positive charge on this molecule will attract it to anything of negative charge in the cell, like the mitochondria here, that's loaded with negative charged electrons, which is the powerhouse of the cell. The only trouble is, this molecule can't decide whether it might go here or might go to the left here where we have DNA, which also has a net negative charge. And so these ring-shaped structures are capable of slipping in through the rungs of the DNA ladder, and then when the DNA starts to unzip to copy itself and comes to where it's lodged, it breaks, and we call that a mutation. I also want to point out that this particular ring-shaped structure is the base structure for atrazine, which is a very common herbicide and is applied generously in crops that are resistant to it. We'll see about that a little bit more. So we decided, we were curious to know whether or not a lawn chemical might be capable of having biological activity, since when you look at the ingredients in a very common herbicide, this happens to be a mix that is what Trimec has in terms of its ingredients. This was 2,4-D right here that we've been talking about a lot, a ring-shaped structure, fat solubility, negative acid group, a negative chloride, so it's got water solubility, fat solubility. Same for mecoprop. Same for dicamba. I also want to point out, and this is very important, that every 2,4-D that comes off the shelf is going to have at least 2 small dioxins in it. It's part of the process of manufacture. It's inevitable. And so when you put it on your lawn, or you have GMO foods that have it, or have this used on it, you've got dioxins in there. Why are they important? Because they are the most potent estrogenic kind of pseudoestrogen we have on the planet. They bioaccumulate, and they have numerous neurological, endocrine, immune, developmental impacts. We're going to talk about some of that. So here's a reminder. The ring-shaped structure gives you fat solubility. Negative charge or positive charge gives you water solubility. Whoops. So we decided, okay, let's take a look at herbicide induction of abortions and resorptions of embryos. And is there a neuroendocrine immune effect? So this is work we published in 2002 in Environmental Health Perspectives. We decided we'd dose the drinking water of pregnant mice with diluted herbicide, and we do it either days 1 through 15 through the end of organ formation, or we would do it from day 5 to day 15. We decided we'd focus on 2,4-D and let the other 2 come along for the ride in terms of concentrations. So we picked an intermediate concentration here in terms of milligrams per kilogram per day, 20. EPA said maybe there's a little bit of effect here, but EPA doesn't look at this kind of thing. We also chose a high dose, and then we went to a low dose and a very low dose down here. This is 39 parts per billion here. Okay, so we wanted to know, do we have embryo losses? And how do you do that? You measure the number that were implanted minus the number that are born. Difference is what are lost. These are 2 mouse uteri. What we do is we let the pregnant mom, bear her young. The young are born. She nurses them. They're weaned, and then we remove their uteri, and we put them in ammonium sulfide stain. What that does is every place where and the lights are up, I don't know if you can see this, there's a black spot, another black spot, another black spot. These are each where there was a placenta implanted on the last pregnancy. This is the cervix. This is where the fallopian tubes come in on each one of the horns of the uterus. These are like peas on a pod. You can just count them. So you know how many attachments there were, how many mice, how many babies there were in the uterus. When we take a look at the results from this experiment that we published in 2002, we have results here. The average litter size, about a little over 10 for the control animals that got nothing. And here's the high dose, the intermediate dose, the low dose, the very low dose. Somebody please tell me which of those treatments had the greatest fetal losses? The very low dose. The greatest effect was at the lowest dose. And here are the actual numbers. Here are the litters, a number of litters that we had at different litter sizes. Smallest litter size was 8 here in the controls. Here are litter losses. Litter losses in the dark colors, more litter losses. Every single dose had losses, but the greatest effect was at the lowest dose. I want to point out that atrazine, with its ring-shaped structure, also has reproductive effects. Tyrone Hayes at Berkeley has been looking at frogs, and when he looks at control males, their fertility is up here pretty close to 100%. But if you put them in environmentally relevant atrazine levels that you'll find outdoors, here's the fertility for atrazine-treated males. More importantly than that, if we look at the behavior of the frogs, these are two male frogs trying to copulate with each other. And Syngenta, Tim Pasteur, the representative guy from Syngenta, says at this time EPA believes that no additional testing is warranted to address this issue. Syngenta, by the way, also makes tamoxifen, which is the chemical used in breast cancer, which is driven also by estrogenic properties. Now, why do we know that atrazine improves estrogen concentration or enhances it? Because there's a phenomenon where testosterone is converted one way to estrogen by way of this enzyme aromatase. And what atrazine does is it increases the amount of aromatase, so you get more testosterone converted to estrogen, you elevate your estrogen. You feminize, which means you lower sperm production in males. You get ovaries and testes in the same animal. You get mammary tissue in male breasts. Roundup, on the other hand, which is our other big GMO product, will do the opposite. It tends to shut down aromatase, build up testosterone, and so if you have a female fetus, you can masculinize it, apparently, in utero. So we have two of these very common herbicides that are capable of altering the key sex hormones that determine sex during embryonic development. Now the question is, might estrogen changes alter human sexual behavior? The estrogen receptor, we know, is very promiscuous. It has at least 12 different partners that it will bind with. Bruce McEwen, in Environmental Health Perspectives in 1987, published a very interesting article with this particular title here, and I want to go to the next page, which is part of his paper, where he talks about psychosexual differentiation and diethylstilbestrol in humans that were exposed in utero. This is a pseudoestrogen. And he says, in studies thus far completed and published, prenatal diethylstilbestrol, a pseudoestrogen, alters general measures of personality and leads to altered patterns of sexual behavior in adolescence and adulthood that reduce formation of heterosexual relationships. These differences from carefully matched normal subjects could not be explained by sexual dysfunctions such as vaginismus and dyspareunia, which were low in both groups, but rather appear to be due to psychosocial and neuroendocrine factors related to this diethylstilbestrol exposure in utero. And here are the papers that they cite. I want to point out that 2,4-D is always containing dioxins, which are potent xenoestrogens. This was a paper by Meg Sears, and what she wrote in this paper that was published in 2006 was way over here. And basically what she says is that dioxins are formed during the manufacture of this compound with higher chlorinated congeners and furans being produced at increased temperatures. Normally, dioxins with 2 or 3 chlorine atoms are formed in the manufacture of 2,4-D, although higher chlorinated congeners have been measured in Canadian products. Higher reactor temperatures also favor more rapid and complete conversion of reactants to herbicide during manufacture, so that there's concern that in the absence of enforced regulatory limits, the economic incentive to improve efficiency may foster the production of higher contaminated herbicides. But basically, they are more dangerous and bigger with trying to speed things up. In 2009, we had a paper showing that dioxins can alter sexual behavior in both sexes of rats. And I know I'm just about out of time. I just want to summarize what we know now, and that is that current data from animals and humans suggest hormonal, neurological, and immunological effects of herbicides at environmentally relevant doses. And if you'd like to see more about that, here's a talk I gave about a year ago that's on the web. Second thing is that current data indicate that common herbicides and their dioxin contaminants are likely assaulting sexual development and sexual behavior in animals and humans. Now the question is, will our society continue to sexually assault our children in and out of the womb or abort those fetuses in the name of weed-free lawns and crops? We don't allow sexual assault in our churches. Why should it be legal in our homes, yards, and croplands? We have MAD, Mothers Against Drunk Drivers, but it seems to me we need something like MAM, Mothers Against Molestation. Thank you very much.
Thank you very much.